Hello, very good evening to the student who have joined. Can you please let me know in the chat box if my voice is audible to you? Again, let me know, please, if the PPT is visible. In the chat box, kindly let me know if you can hear my voice properly and if you can see the presentation. Okay, thank you so much. We will wait till 6 p.m. and then we will start today's session. Meanwhile, let me know if there is any issue with the audio and also with the PPT because due to the poor internet connection. So we will wait till 6 p.m. and then I will start today's session. Okay. Meanwhile, please let me know if my voice is audible to you and also if there is any issue with the audio and presentation, kindly let me know. Okay. Okay, I will make the call open and I will start the session. Okay, I think I can start the session now. So, very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this course, NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is our week 8 live session and here we will discuss the course content of week 9. So every week we are going ahead with a course. This in the our first live session we covered week 1 and week 2 all together. So subsequently today we are going to cover the week 9 live course content which is live on the SWAYAM portal of NPTEL. Okay, every week we have this kind of live session where we discuss around 20 MCQ question which is related to that week's course content. All these questions are MCQs and there will be 4 options. You have to select the correct answer and you have to write it in the chat box. I will check the answer and then I will discuss that particular topic. In this way, you will be accustomed with solving the MCQ questions which are in your weekly assignments, and also you will understand the course better. So before starting, I would like to cover a few points. So there will be a problem solving sessions every week. Okay, these sessions will be live on Google Meet platform, and the sessions will be on every Monday from 6 p.m. to 8 p.m. So if any network disruptions happen, please wait for a few minutes. Do not leave the session. I will try to join as soon as possible. Okay, during the session, students can post their queries, their doubt, their questions through the chat box. Since I am recording the session, okay, and for the smooth recording, I have muted you all and closed and kept your cameras off so that I can record the session without any problem. This recording I will share to you later after the class. So if you have any doubt, any query, please let me know in the chat box. So the chat box is always open. I am monitoring the chat box. So any of your doubts and queries, you can let me know through the chat box. Okay, if there are any network disruption from my side, please do not leave the class. Particularly for today, the weather is very poor today here. So that is why there might be some network disruption. So I request you beforehand that please do not leave the class. I will try to join as soon as possible. Okay. And after the class, if the doubt persists, I will drop my email ID in the chat box. You can let me know your queries through the email also. And this presentation and class recording will be shared with you through the SWAYAM portal. So in the SWAYAM portal, where there is a assignment tab, there you will find a problem solving session tab also. In that problem solving session, if you go, you will find that particular week's recording and also the feedback. Okay, you can give your feedback also so that I can understand that the things I need to cover more in your feedback according to your feedback. So I think today it is our week 8 session and we are going to cover a week 9 course content. Once I show you one question, I will give you some time. I will give you 2 to 3 minutes time. You read the question carefully, okay, you understand, and then let me know in the chat box that A, B, C or D, which of the following answers will be the correct one. Okay, and then you let me know that which of the following answer will be the correct one. I will check your answer and then what I will do is I will show you the correct answer and we will discuss the topic thoroughly. That is how we will understand the particular topic also, and you will identify how to solve your assignment. You will you will know how to solve your assignments. Okay. So let us start with the session. Meanwhile, when the more students join, they can join accordingly. Again, I am saying there is a very network disruption might happen today because of the poor weather, but do not leave the class. If there is any network issue from my side, I will try to rejoin as soon as possible. Okay, so do not leave the session. So let us first see what are in the course content of week 9. So week 9 course contents are like this. The first is the introduction to drug discovery process. Then we have fundamental principles of drug development process. We have combinatorial chemistry, neurotransmitters, catecholamine based and GABA neurotransmitters, hypertension, humoral mechanism and renin-angiotensin system, and inhibitor design of angiotensin converting system. So these are the topics of week 9 course content, and I will give you questions regarding to these topics today. Okay. So now let us first go to the first question of today's session. So problem number 1, it says that which of the following reflects the order in which various drug discovery and development stages take place. Your options are: number A, target identification; establishing a bioassay, finding a lead, and SAR. Number B, establishing a bioassay, target identification, finding a lead, and SAR. Number C, target identification, establishing a bioassay, SAR, and finding a lead. And number D, target identification, SAR, establishing a bioassay, and finding a lead. So which of the following reflects the correct order in which various drug discovery and development stages takes place? I will give you some time. Think and let me know in the chat box A, B, C or D which of the following will be the correct answer. I will wait for 2 to 3 minutes. A, B, C or D which of the following will be the correct answer for this problem? Let me know in the chat box. Let me check the chat box. Okay, I have got few questions. Okay, if you have any doubt, please let me know in the chat box. Okay, C, D, D, A. So I have 4 answers. Number C, number D, number D, and number A. Where means number C, D, and A. 3 options are said to be correct. Okay, let me check which of the following will be the correct answer. So the correct answer is number A, target identification followed by establishing a bioassay, finding a lead, and then SAR is the correct order that reflects the drug discovery and development stages. So this is your series of drug discovery and development. So first is the market analysis. Okay, the input from different. So first is the market analysis, which gives us the target identification. Okay, the target before the target identification, we have this UMN identification. UMN means unmet medical need. Okay, this comes from the market analysis input from different KOLs and practitioners. Then we have the first step, which is the target identification, which comes from the phenotypic screening, genetic association, transgenic organisms, and imaging, followed by target validation by antisense and RNAi technology, genetically modified animals, and proteomics. Then we have the heat identification. It comes from the assay development. HTS means high throughput screening, fragment based screening, and focus screening. Then followed by we have the counter heat confirmation by counter screening of dose response and functional assay. Then followed by the lead generation, which comes from the conventional and combinatorial chemistry. Then after that we have the SAR establishment, activity and selectivity, physiochemical properties, ADME, pharmacokinetics, toxicity potential, in vivo PK, primary POM, disease models. Then we have this lead optimization, we enhancement of physiochemical and ADME properties, PK-PD correlation, metabolic ID and characterization, backup medicinal chemistry strategy, followed by the preclinical study. So let us understand, first is the target identification. You have to first identify what is the target, what is the disease. First you identify the disease, then you have to identify the target. Now if for a particular disease there can be many targets, like for cancer there are various targets which can be used to develop an anti-cancer drug. Okay, because there are various enzymes, there are various receptors, and so on. Okay, various cell cycle pathways. So there are different targets. So first thing is the target identification. After identifying the disease, you have to identify the target. Then you have to establish a bioassay. So establishing a bioassay means establishing a process or method through which you will find a particular lead or a particular lead compound. Then it will be subjected to SAR modification, which are structure activity relationship modifications by and medicinal chemistry, followed by the preclinical candidate. Okay, it will give us a preclinical candidate. So correct order was number A, target identification, establishing a bioassay, finding a lead, and SAR. Okay, so number A was the correct answer, and I think there was a number A, option number A in the chat box also. Okay. So very good. Others also very good because you have tried. Everyone is answering, so which is very important. Now I will move toward the next problem. Problem number 2 says, which of the following statements is true? Your options are number A, drugs and drug targets have similar molecular weight. Number B, molecular weight of the drug target is higher than that of the drug. Number C, molecular weight of the drug is higher than the drug target. And number D, there is no general rule regarding this. Again, I am repeating, which of the following statements is true? Number A, drugs and drug targets have similar molecular weight. Number B, molecular weight of the drug is higher. Molecular weight of the drug target is higher than that of the drug. Number C, molecular weight of the drug is higher than that of the target. And number D, there is no general rule regarding this. Again, I am giving you some time and identify A, B, C or D, which of the following is the correct answer. Which of the following statement is true? Let me check the chat box. Yeah, the doubt solving recording will be available till the date of the exam. It will be available all the time. You don't have to worry about it. You will find on the SWAYAM portal there is a link. It will be active throughout your course, okay? Till the examination. There is no problem. Okay, I think I have few answers for the question numbers. The second question, number B, number A, number B, number D. Number A, B, and D are three different answers which I have got for the problem number 2. Let me check if which of the following is the correct one. So the correct answer is number B. Molecular weight of the drug target is higher than that of the drug. This statement is true. Okay. So what is a drug? So basically a drug is a chemical substance, a natural or human made, that changes the normal body functions in some way. So there is no set of definition of the term drug, but this is one we will use in the class. So drugs are used to cure or treat disease, reduce symptoms of illness, alter a person's mental or physical state. So this is the chart, okay, the pie diagram. So there are different drug targets, like enzymes comprises of 47% of drug target. We have DNA, we have integrin, miscellaneous, other receptors, GPCRs, ion channel transporters, and nuclear hormone receptors. So these are the various drug targets for which we can, we can find out a drug. So basically the molecular weight of a drug target is always should be higher than the drug. Like for example, enzymes or any kind of DNA or these things, these are made of what mainly is the protein or amino acids. Okay, like enzymes are made of the protein. The molecular weight of the enzymes are definitely very high with respect to the drug. Okay, because the drug should not have the molecular weight most greater than 500 daltons. So there is a rule regarding that, which is very famous rule of Lipinski's rule of 5, which says that a compound to be good viability of a drug, it should not have more than 5 hydrogen bond donor, not more than 10 hydrogen bond acceptors. The molecular weight should be less than 500 and C log P not greater than 5, which is the value of the lipophilicity. These are the physicochemical properties of the which comes from the Lipinski's rule of 5. Okay, so the statement number B, which says molecular weight of the drug is should be the higher than that of the drug, it is the correct statement. Okay, number B. Now I will go to the next problem. So problem number 3 says in drug discovery process, pharmacokinetics is a study is done during. Your options are number A, basic research, number B, post-market surveillance, number C, preclinical study, and number D, clinical development. So which of the following is the correct answer? In drug discovery process, pharmacokinetic study is done during number A, basic research, number B, post-market surveillance, number C, preclinical study, and number D, clinical development. Let me check the chat box now. Okay, I have only one answer as only on one on option as answer, which is the number C. Let me check if it is the correct one or not. So the correct answer will be very good. It is number C. During the preclinical study, the pharmacokinetic study is done. So basically, as I have already I have discussed that first is the disease target identification followed by the basic research. So now what is the basic research is the target identification, target validation, assay development, genomics and proteomics. Then we have the early discovery, we have the computational studies, high throughput screening, identification of hit to lead and lead optimization. Then we have the preclinical studies where we have our in vitro, in vivo studies, pharmacokinetics formulation and drug and dosage studies. Then we have the clinical development followed by the FDA approval, and then we have the post-market surveillance. So these are each are the one or each part of the drug discovery development process. Okay, so in the basic research, we add after identification of target and target validation, we have the assay development. Then early discovery, we have the different computational study where we identify there are different leads and lead to hit identification. Preclinical studies, we will have to do the in vitro, in vivo, all the data sets. We have to generate the pharmacokinetics and SARs, and then clinical development followed by the FDA. So FDA is the Drug Development and Food and Drug Development and Administration of USA, which after approval of FDA, any drug can come to the market. After the market, in the market, the drug stays in the market for 1 to 2 years. Then we have to check the post-market surveillance where we have to identify that if there is any long-term side effect of that particular drug or not. Okay, so these are the complete drug discovery and development process. Now problem, so what is the now pharmacokinetics? Pharmacokinetics is basically what is our body is doing to a particular drug. So it is also known as the ADME. Okay, so ADME, what is it? It is the first is the administration when the dose of drug is administered, which is the input, followed by the distribution of the drug concentration in the systematic circulation. The distribution means drug in the tissues of distribution, followed by the drug concentration at the site of the action. The drug is metabolized or excreted is the elimination. Then we have the pharmacologic effect, clinical response, toxicity and effectiveness. This first portion is the pharmacokinetics and this is the pharmacodynamics, which means pharmacodynamics means what this that particular drug is doing to our body. So the different pharmacological effect, the clinical response, toxicity, effectiveness, all are this part of the pharmacodynamics. Okay. Now I will go to the next problem. So problem number 4 says there are several sources and methods of discovering new compounds. Which of the following is most likely to lead to the discovery of a complex structure quite unlike any other previously discovered? Your options are number A, combinatorial chemistry, number B, database mining, number C, natural product screening, and number D, me too drugs. There are several sources and methods of discovering new compounds. Which of the following is the most likely to lead to the discovery of a complex structure quite unlike any other previously discovered? Your options are number A, combinatorial chemistry, number B, database mining, number C, natural product screening, and number D, me too drugs. Okay, let me check the chat box. Okay, I have different answer. I have one number C and other I have number A. Okay, let me check which of the following is the correct answer. So the correct answer is, it is number C. Natural product screening is the most important way of having the different new compound of a very complex structure unlike previously discovered. So natural products are rich source of biologically active compounds. Many of today's medicines are either obtained directly from the natural source or were developed from a lead compound originally obtained from a natural source. Usually the natural source has some form of the biological activity and the compound responsible for that activity is known as the active principle. Such a structure can act as a lead compound. Most biologically active natural products are secondary metabolites with quite complex structure and several chiral centers. This has an advantage in that they are extremely novel compounds. Unfortunately, this complexity also makes their synthesis difficult and the compounds usually have to be extracted from their natural source. A slow, expensive and ineffective inefficient process. As a result, there is usually an advantage in designing simpler analogs. Many natural products have a radically new chemical structure which no chemist would dream of synthesizing. For example, antimalarial artemisinin, a natural product which is an extremely unstable looking trioxane ring, one of the most unlikely structures to have appeared in the recent years. The study of medicines derived from the natural sources is known as pharmacognosy and includes the crude extracted and purified active principles. So there are different natural products which can be screened at the plant kingdom, microorganism, marine products, animal sources, venoms and toxins. Okay. So natural product screening was the correct answer. Now I will go to the next problem. So problem number 5 says which of the following is not derived from the microorganisms? Your options are number A, cyclosporin, number B, curaxin A, number C, lipstanine and number D, rasponin. Which of the following is not derived from the microorganisms? Let me check the chat box now. Okay, I have, I think, two answers, which is number A and number B. Let me check if which of the following is the correct one. So the correct answer is number B. Curacin A is the compound which is not derived from the microorganism. So microorganisms such as the bacteria and fungi are also provided rich pickings for the drugs and lead compounds. These organisms produce a large variety of microbial agents which have evolved to give their host an advantage over their competitors in the microbiological world. The screening of the microorganisms became highly popular after discovery of penicillin. Soil and water samples were collected from all around the world in order to study new fungal and bacterial strains, leading to an impressive arsenal of the antibacterial agents such as the cephalosporins, tetracyclines, aminoglycosides, rifamycins, chloramphenicol, and vancomycins. Although the most of the drugs derived from microorganisms are used in antibacterial therapy, some microbial metabolites have provided lead compounds in other fields of medicine. For example, asparagine, isolated from Aspergillus aureus, a noble antagonist of the peptide hormone called chlorocytokinin, which is involved in the control of appetite. CCK also acts as a neurotransmitter in the brain and is thought to be involved in panic attacks. Analogs of asparagine may therefore have potential in treating anxiety. Other examples include the fungal metabolite lovastatin, which is the first clinically useful statins found under lower rather cholesterol levels, and another fungal metabolite called cyclosporin, which is used to suppress the immune response after organ transplant. Lipostatin is a natural product which is isolated from Streptomyces toxicrocini. It inhibits pancreatic lipase and was lead compound for the anti-obesity compound orlistat. Finally, a fungal metabolite called rasporin, which is isolated from the fungal, promotes the cell death in apoptosis, that's cancer cells, but not normal cells. It represents a promising lead compound in novel anti-cancer agents. So these are the structures for lead compounds from the microbacterial sources like rasporin, cyclosporin, and lipostatin. Then we have the marine sources. In recent years, there has been a great interest in finding the lead compound from the marine sources. The coral, sponges, fish, and marine microorganisms have a wealth of the biologically potent chemicals with interestingly inflammatory, antiviral, anti-cancer activity. For example, curacin A, which has this structure, is obtained from the marine cyanobacterium and shows potential anti-tumor activity. Other anti-tumor agents derived from marine sources include the eleutherin, virestatine, dolastatin, cephalostatins, and helicondrin B. In 2010, a simplified analog of helicondrin B was approved for the treatment of breast cancer. Okay. So, curacin A is the compound which is not derived from the microorganism but from the marine sources. I will go to the next problem. Problem number 6 says identify the correct statement about the combinatorial chemistry. Your options are number A, one molecule can be done at one time. Number B, it has high risk of failure. Number C, it was developed using solution phase synthesis. And number D, it creates an impact in lead discovery and optimization. So which of the following statement is correct regarding combinatorial chemistry? Your options are number A, one molecule can be done in one time. Number B, it has high risks of failure. Number C, it was developed using solution phase synthesis. Number D, it creates an impact in the lead discovery and optimization. Let me check the chat box now. Okay, I have few answers. Number C, number D, number C. Okay, I have number C and number D are the most given answer. Let me check if the which of the following is the correct one. So the correct statement is number D. It creates an impact in lead discovery and optimization. So combinatorial chemistry is a technique by which large numbers of different but structurally similar molecules are produced rapidly and submitted for pharmacological assay. This technique uses same reaction condition with the same reaction vessels to produce a large range of analogues. Technique invented in late 1980s and early 1990s to enable tasks to be applied to many of molecules simultaneously. So combinatorial library that from the products produced from combinatorial synthesis are known as combinatorial library. Libraries may be a collection of individual compounds or mixture of compounds. Screening and of the components of the library for activity using high throughput screening techniques enables the development team to select the suitable compounds. This is the combinatorial chemistry within drug research. First is a therapeutic agent, followed by lead discovery, lead optimization, development of the candidate, and drug. So combinatorial chemistry can make impact in the lead discovery and lead optimization. So conventional drug discovery, one molecule can be done at one time. In combinatorial, many molecules can be done at one time. The convention is a make, purity, and test. Now, combinatorial make, test, and purity. Hundreds of molecules can be done in months in conventional, and thousands of molecules can be done in a month. Slower lead generation is in conventional and combinatorial, faster lead generation. High risk of failure in the conventional and low risk of failure in the combinatorial, which gives us the synergy and the lead identification. Okay, right, it creates an impact in this discovery and lead optimization. Now, problem number 7 says solid phase synthesis is frequently used in combinatorial chemistry. What is meant by solid phase synthesis? Your options are number A, reactions are carried out without a solvent. Number B, reagents and reactants are attached to a solid phase support. Number C, reagents are used in solid phase. Number D, molecules are constructed on a solid phase support. So, which of the following statement is correct regarding the solid phase? What is the mean by solid phase synthesis? In the chat box, let me know A, B, or C, or D, or which or A, B, C, or D, which is the following is the correct answer. Let me check the chat box now. Okay, I have few answers. Number B and number D are said to be the correct answer. Let me check which of the following is the correct one. So the correct answer is number D. Molecules are constructed on a solid phase support. This is made by the solid phase synthesis. So now, what is the solid phase synthesis? Combinatorial synthesis is based on solid phase chemistry. It uses filtration as a separation and purification technique. The easy removal of unwanted substance by a simple filtration is the core of the library synthesis. In this synthesis, substituted resin beds are used as a solid phase, which is actually a gel-like matrix of connected polymeric molecules distended by the axis of the solvent molecules. The use of the resin beds, the use of the solid support for organic synthesis requires a cross-linked insoluble but solvent soluble polymeric material that is inert to the conditions of synthesis. Some means of linking of the substrate to this solid phase that permits the selective cleavage of the product from the solid support during the synthesis. A synthetic procedure compatible with the linker and the solid. Okay. So these are the solid phase which is the require of the solid support. Now I will go to the next problem. So problem number 8 says the term used to signify the biological effect of a preparation without the active drug that appears to induce a beneficial effect similar to that of the active preparation is number A placebo, number B agonist, number C me too drugs and number D peptidomimetic. So which of the following is the correct? The term used to signify the biological effect of a preparation without the active drug that appears to induce a beneficial effect similar to that of the active drug preparation is number A placebo, number B agonist, number C me too drug and number D peptidomimetic. Identify the correct term. Let me check the chat box. Okay, I have few answers. Number A, number C, number B. Let me check which of the following is the correct one. So the correct answer is number A, placebo. Okay. So placebo is a substance that has no known specific pharmacological activity against the condition being treated. Placebos can be therapeutically beneficial to some patients when they give rise to the placebo effect. So what is the placebo effect? A fake treatment can make someone better simply because they expect it to help. Okay, so placebo is can be real or can be fake. It can be a medicinal or a normal compound. Okay, but since as long as the patient believes that it is a drug and it will make him or her better, it will act. Okay, so this is known as the placebo effect. So now let us understand the other terms also. What is the agonist? So agonist is the drugs that occupy receptors and activate them. Antagonists are the drugs that occupy the receptors but do not activate them. Antagonists block the receptor activation by agonist. So me too drugs, a drug that is structurally very similar to already known drugs with a minor differences is the term me too carries a negative conclusion. However, me too products may create the competition to drive the prices down. Me too drugs can be broadly defined as a chemical related to the prototype or other chemical compounds which have identical mechanism of action. Peptidomimetic, incorporating conformational constraint locally or globally. What is peptidomimetic? These are molecules with significantly reduced peptide character that mimic the bioactive conformation of peptides, thus retain the ability to interact with the biological target and cause the same biological effects. Small protein like chain designed to mimic peptide arise either from modification of an existing peptide or by designing similar systems to mimic the peptide. For example, peptoids or beta peptides. Okay. Now I will go to the next problem. Problem number 9 says which of the following methods can be used for lead design? Your options are number A IR, number B NMR, number C ESIMS and number D GCMS. Which of the following methods can be used to for lead designing? IR, NMR, is ECIMS or GCMS? Let me check the chat box. We have a few answers. Number A and number B are the given answer. Let me check which of the following is the correct one. So the correct answer is it's number B, NMR. NMR is the method that can be used for lead design. So recently, NMR spectroscopy has been used to design a lead compound rather than to discover one. In essence, the method sets out to find small molecules or epitopes which will bind to specific but different regions of the protein's binding site. These molecules will have no activity in themselves as they only will bind to a part of the binding site. But if the larger molecule is designed which links these epitopes together, then a lead compound may be created which is active and binds to the whole of the binding site. So this is the binding site of the protein. The first screen for the first ligand, the protein can bind here, optimization, and then screen for the second ligand, it will bind to this portion, and then the linkage can be formed between these two leads. Okay, this is known as the epitope mapping. So the use of NMR spectroscopy in finding lead compounds. NMR spectroscopy was used to design high affinity ligands for the FK506 binding protein, a protein involved in the suppression of the immune response. Two optimized epitope A and B were discovered, which were bound to the different regions of the binding site. Structure C was then synthesized whether the two epitopes were linked to the propyl link. This compound have higher affinity than either of the individual epitopes and represents the lead compound for their further development. Okay, so this is the epitope A, this is the epitope B, and they go together form makes the particular design of a ligand. Okay, and this linkage was, so this is the epitope A which is here, epitope B which stays here, and this linkage is formed later once the particular two leads are bind to a protein. After that the link can be happen, and this ligand is designed by use of the NMR technique. Okay, so the NMR was the correct answer. Now, problem number 10, it says, which of the following statements best describes pharmacokinetics? Your options are the study of how drugs reach their target in the body and how the levels of the drug in the blood are affected by various factors. Number B, the study of how drugs can be designed using molecular modeling based on the drug's pharmacophore. Number C, the study of how the drug interacts with its target binding site at the molecular level, and number D, the study of which functional groups are important in binding a drug to its target site and identification of a pharmacophore. So which of the following statement best describe the pharmacokinetics? Let me check the chat box. Okay, I have few answers. Number A, number B are the given options. Let me check which of the following is the correct one. Okay, so the correct answer is number A, the study of how the drugs reach their targets in the body and how the levels of the drug in the blood are affected by various factors. So it is describes the pharmacokinetics. So pharmacokinetics is the movement of the drug through the body from entrance to exit. Now, the absorption is the movement of the site of administration to bloodstream. Common routes of administration are oral, intravenous, intramuscular, subcutaneous, transdermal, and pulmonary or nasal. Bioavailability is the extent or rate of which the drug enters the systematic circulation. Bioavailability is 100% in the IV administration but varies widely in other routes. Distribution, transport through the systematic circulation to target tissues or organs influenced by adequate blood flow, ability of the drug to exit the vascular system, ability of drug to enter the target cell. Factors that may limit the distribution is the vascular damage due to the chronic disease such as diabetes, limited blood supply to abscess or tumors, tight junctions of the blood-brain barrier. Metabolism, clinical chemical alteration and biotransformation of the drug to make the pharmacologically active to facilitate elimination primarily occurs in the liver. Effect of metabolism may vary by drug, may include the conversion of the lipid soluble to water soluble compound, inactivation of drug, bioactivation of prodrug, increased or decreased toxicity, excretion, most directly filtered by kidneys and by, for example, the bile and by feces. Okay. So, pharmacokinetics is the study of how the drugs reach to the target and how the levels of the drug in the body are affected by various factors. And pharmacodynamics is how the drug is affecting our body throughout the various mechanisms. Now, problem number 11 says the incorrect statement among the following is number A, lesser the value of ED50, the drug will be more potent. Number B, larger the therapeutic index, safer is the drug. Number C, therapeutic index doesn't depend on ND50. And number D, lower is the IC50, the better is the drug molecule. So, which of the incorrect statement among the following is? Number A, lesser the value of ED50, the drug will be more potent. Number B, larger the therapeutic index, safer is the drug. Number C, therapeutic index doesn't depend on ND50. And number D, lower is the IC50 value, better is the drug molecule. Let me check the chat box now. I have answer C, answer C. Okay, let me check if answer C is the correct one or not. Yes, answer C is the correct one. Therapeutic index doesn't depend on LD50. So the therapeutic index is the ratio between the doses of the drug that causes a toxic or lethal effect and the doses that causes a therapeutic effect. So therapeutic index is TD50 by ED50 in case of humans, and in TD50 means the median toxic or lethal dose, and ED50 is the effective dose. So TD50 is the toxic dose for the 50% of the population, and ED50 is the effective dose for the 50% of population. Okay, in case of humans. In animal, it is LD50 by ED50, where LD50 is the lethal dose, which for the 50% of population. So therapeutic index doesn't depend on LD50. This statement is incorrect. Okay, the lesser the ED50 value, the drug will be more potent. It is the correct one. Larger the therapeutic index, safer is the drug. Again, the correct one. Therapeutic index doesn't depend on LD50. This is not correct, because as you can see from the formula, it greatly depends on the ED50, LD50, and lower the IC50 value, better is the drug molecule. And is also the correct answer. Okay. IC50 means the inhibitory concentration at 50%. Okay, so to inhibit a particular drugs, if a biological action to 50% or inhibit a particular enzyme effect to 50%, the concentration of drug needed is known as the IC50 value. The lower the IC50 value means lower the particular drug is required to inhibit the particular target, so it is a better drug. Problem number 12 says, which of the following statements regarding neurotransmitter is true? Your options are: number A, they are secreted by the endocrine glands. Number B, glutamate is a type of neurotransmitter. Number C, they traverse through the blood to act on a distant target cells. And number D, their effects are longer lasting. So which of the following statements regarding the neurotransmitters is true? Your options are: number A, they are secreted by the endocrine glands. Number B, glutamate is a type of neurotransmitter. Number C, they traverse through the blood to act on a distant target cell. And number D, their effects are longer lasting. Okay, let me check the chat box now. Number C, number B, number A. So I have three different answers, number A, B, and C. So let me check which of the following is the correct one. So the correct answer means the statement which is true regarding neurotransmitter is number C. Glutamate is a type of neurotransmitter. It is the true statement. So neurotransmitters are the chemical substance released by the electrical impulses into the synaptic cleft from the synaptic vesicles of presynaptic membrane. It then diffuses to the postsynaptic membrane, binds to and activates the receptors present, leading to the initiation of new electrical signals or inhibition of the postsynaptic neuron. Different neurotransmitters are adrenaline, which has a fight or flight. It produced in the stressful situation increases the heart rate and blood flow, leading to the physical boost and heightened awareness. GABA or gamma amino acid, butyric acid, which calms the firing nerves in the central nervous system. High levels improve focus, low levels cause anxiety. Also contributes to motor control and vision. Noradrenaline is concentration, affects the attention of the responding actions in the brain, contracts the blood vessels, increasing the blood flow. Acetylcholine is the learning involved in thought, learning, and memory, activates the muscle action of the body, also associated with the attention and awakening. Dopamine is the for pleasure, feeling of pleasure, also addiction, movement, motivation. People repeat behaviors that lead to dopamine release. Glutamate is the memory, most common neurotransmitter involved in learning and memory, regulates development and creation of nerve contacts. Serotonin in swarm mood contributes to well-being and happiness, has sleep cycle and digestive system regulation affected by exercise and light exposure. Endorphin is for euphoria, released during exercise, excitement, and producing well-being and euphoria, reducing the pain. So there are difference between the hormones and neurotransmitter. In some cases, they may seem to be very different, very similar, but these are very different. For example, hormones are released by the endocrine glands. Neurotransmitters are released by the nerve cells. Hormones are travel through the blood to act on a distant target cell. Neurotransmitter travel across the synapse to act on the nearby cells. Hormones, the effects of the hormones can be slower built but longer lasting. Effects of the neurotransmitters can be fast act but shorter lasting. Hormones are part of the endocrine system. Neurotransmitters are part of the nervous system. And this is the neurotransmitter. So this is the nucleus and this is the cell. These are the receptor and this is the neuron which gives the signal to the messenger. And this receptor and the messenger activation produce a response. So that is how neurotransmitter act as chemical messengers that bind to receptors and trigger the reactions within the cell. Problem number 13 says levodopa is used as a prodrug for dopamine since it enters the brain more effectively than dopamine. Why? Your options are number A, it is more polar and crosses the blood-brain barrier effectively. Number B, it is less polar, crosses the blood-brain barrier more effectively. Number C, it mimics an amino acid, takes advantage of the carrier proteins in order to cross the blood-brain barrier. And number D, it is less susceptible to metabolism. So which of the following statements is correct? Levodopa is used as a prodrug for dopamine since it enters the brain more effectively than levodopa. Why? Number A, it is more polar and crosses the blood-brain barrier more effectively. Number B, it is less polar and crosses the blood-brain barrier more effectively. Number C, it mimics the amino acid, takes advantage of carrier proteins in order to cross the blood-brain barrier. Number D, it is less susceptible to metabolism. Let me check the chat box. Okay, problem number 12, answer B is correct. Okay, let me check in problem number 12 again. Oh, okay. So, okay, the option B is correct one. Okay, this it is this different, but yeah. As you can see, I have given the number answer B as the answer, but only the numbering was incorrect. Thank you for pointing out. Okay, now let me check the answer now for problem number 13. Correct answer is number C. It mimics an amino acid, takes advantage of carrier proteins in order to cross the blood-brain barrier. Okay, for the levodopa. So basically, in case of as you can see for levodopa, it mimics the tyrosine. Okay, and then it enters the blood-brain barrier. Okay, it mimics an amino acid. The amino acid that mimics is the tyrosine, and after taking the advantages of the carrier proteins, it crosses the blood-brain barrier and goes to the pro-synaptic terminal from the substance nigra and followed by the dopamine release. Okay. So what is the blood-brain barrier? It is formed by a single layer of endothelial cell that line the inner surfaces of the capillaries. So in the brain, it is a semi-permeable capillary membrane. That is, it allows some materials to cross but prevents others from crossing. In the most parts of the body, the capillaries are lined with the endothelial cells. The endothelial tissue has small spaces between each individual cells, so substances can move readily between the inside and outside the vessel. However, in the brain, the endothelial cells fit tightly together and substance cannot pass out to the bloodstream. Some molecules such as glucose are transported out of the body by the special methods of the active transport. Okay. Now, problem number 14 says, which of the following is true regarding cholinergic receptors? Your options are number A, the inner part of the cell membrane is hydrophilic in nature. Number B, cholinergic receptors are of three types. Number C, nicotinic receptors activate the ligand-gated ion channel opening. And number D, acetylcholine esterase enhances the effect of acetylcholine. Which of the following is true regarding cholinergic receptors? Which of the following statements is true regarding cholinergic receptors? Let me check the chat box. Number C is the given answer. Okay, let me check if number C is the correct one or not. Yes, okay, it is not correct. So the correct answer is number D, acetylcholine esterase or ACHE. Okay, sorry. It is enhances the effect of acetylcholine. Okay. So acetylcholine receptors or ACHRs are of two types. Mainly are the muscarinic acetylcholine receptor or MACHR and nicotinic acetylcholine receptor or NACHR. Nicotinic acid is the two type of neuronal type of nicotinic receptors and muscular type of nicotinic receptors. So acetylcholine receptor, nicotinic receptors fall into three main classes, typically the muscle, ganglionic and CNS type. They are typically a ligand-gated ion channel. Muscle receptors are confined to the skeletal neuromuscular junction. Ganglionic receptors are responsible for the transmission at the sympathetic and parasympathetic ganglia. CNS are three types of the widespread in the brain. Stimulation of autonomic ganglia, stimulation of vulnerary muscle and secretion of adrenaline for the adrenal medulla. Problem number 15 says, which of the following is false regarding the renin-angiotensin system? The options are: number A, aldosterone is a steroidal hormone. Number B, vasoconstriction is caused by the action of angiotensin 1. Number C, aldosterone enhances the retention of sodium and water in the blood. Number D, bradykinin is destroyed by ACE or angiotensin converting system. Which of the following statement is false regarding renin-angiotensin system? Number A, aldosterone is a steroidal hormone. Number B, vasoconstriction is caused by action of angiotensin 1. Number D, aldosterone enhances the retention of sodium and water in the blood. Number D, bradykinin is destroyed by ACE or angiotensin converting system. Let me check the chat box. Okay, I have one answer which says it's number B. Let me check if number B is the correct one or not. So the correct answer is yes, number B, vasoconstriction is caused by the action of angiotensin 1. So this is the renin, angiotensin and aldosterone pathway. First is the dehydration or sodium deficiency or hemorrhage. It gives rise to the decrease in the blood volume followed by the decrease in the blood pressure, which activates the juxtaglomeralar cells in the kidney, which increase the renin. Now we have angiotensin in liver from which increased by the angiotensin 1, and angiotensin 1 is converted by angiotensin 2 by ACE, which are in lungs. It is the angiotensin converting enzyme. Then angiotensin 2 does the two things. One is the vasoconstriction of arterioles, which give rise to the blood pressure increase until it returns to the normal. Again, it gives rise to the adrenal, activates the adrenal cortex, which increase the aldosterone. Then aldosterone in kidneys, the increased sodium and water reabsorption, then increased blood volume followed by the blood pressure normalcy. Okay, so vasoconstriction caused by the action of angiotensin 1, it is the correct one. And this is the aldosterone. So aldosterone has different active aldosterone have different work. For example, for sodium retention or high blood pressure, oxidative stress due to the damage of the DNA or vascular remodeling, for arterial stiffness, renal fibrosis or peritoneal renal functions, cerebrovascular disease, and so on. Okay. Problem number 16 says, which of the following amino acid pairs are removed by angiotensin 1 by ACE? Now, which of the following amino acid pairs is removed from angiotensin 1 by ACE? Number A, histidine and proline. Number B, aspartate and arginine. Number C, histidine and leucine. And number D, valine and tyrosine. Which of the following amino acid pairs is removed by angiotensin 1 by ACE? Let me check the chat box now. Number A, okay, let me check if number A is the correct one or not. The correct answer is, it's number C. Histidine and leucine are the amino acid pair. It is removed by angiotensin 1 by ACE. So angiotensin is converted to angiotensin 1 by the action of renin, and then renin is converted to angiotensin 2 by ACE or angiotensin converting enzyme. So aspartate, arginine, valine, tyrosine, isoleucine, histidine, proline, phenylalanine, histidine, and leucine. This is the 10 amino acid residues in angiotensin 1. ACE cleaves the terminal 2 amino acids from the C terminal, which is the histidine and leucine, and then this is the angiotensin 2. Okay. So angiotensin converting enzyme or AC is the key component of the biosynthetic pathway that generates the hormone angiotensin 2. The pathway involves the conversion of angiotensin to angiotensin 1 catalyzed by the enzyme renin, followed by the conversion of angiotensin 1 to angiotensin 2 catalyzed by ACE. Angiotensin 2 is the potent vasoconstrictor which results in the increased blood pressure, and so drugs that block the synthesis or actions of this hormone can act as antihypertensives. At least there are three main categories of drugs affecting the biosynthesis of action of angiotensin 2: renin inhibitor, ACE inhibitors, and angiotensin 2 receptor antagonists. In the case study, so the design of ACE inhibitors demonstrate how it's possible to design the drugs for a protein protein target in rational manner, even if the structure of the target has not been determined. AC is the membrane-bound enzyme which has been difficult to isolate and study. It is a member of the group of enzymes called zinc metalloproteases and catalyzes the hydrolysis of dipeptide fragment from the end of the decapeptide called angiotensin 1 to give the octapeptide angiotensin 2. Angiotensin 2 is an important hormone that causes the blood vessels to constrict, resulting in rise of the blood pressure. Therefore, AC inhibitors are potential antihypertensive agents because they inhibit the production of angiotensin 2. Although the enzyme AC could not be isolated, the design of AC inhibitor was helped by the studying structure and mechanism of the zinc metalloproteinase that good enzyme called carboxypeptidase. Problem number 17 says the correct statement among the following regarding the antihypertensive drug enalapril is: number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. And number D, all of the above. So which is the correct statement of the among following regarding the antihypertensive drug enalapril? Number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. Number D, all of the above. Let me check the chat box. Number B is the given answer. Okay, let me check. The correct answer is number B. It is a prodrug is the correct statement. Okay. So enalapril is the angiotensin converting enzyme E inhibitor or ACE inhibitor. It's a prodrug which when hydrolyzed by esterase is active from enalaprillate. Mechanism of action: enalaprillate competes with angiotensin 1 for binding at the angiotensin converting enzyme, blocking the conversion of angiotensin 1 to angiotensin 2. The angiotensin 2 is a vasoconstrictor and negative feedback mediator of renin activity. Lower the concentration results in the decrease in blood pressure. Enalaprillate may also act on the kinase 2 that decreases the vasodilator bradykinin. This is the enalapril. It is a prodrug that can cross the membrane. Enalapril is an antihypertensive agent. Then it is the enalaprillate, okay, the structure, and it is the ACE enzyme, where it is the ACE, it is the binding pocket for the, and there is a binding pocket. Okay, the design of the angiotensin converting or ACE inhibitors. Now, problem number 18 says, tell me certain is a dash type of antihypertensive agent. Your options are number A, diurate, number B, beta blocker, number C, ACE inhibitor, and number D, AT1 blocker. So you have to identify, tell me certain is which type of antihypertensive agent. Telmisartan is a dash type of antihypertensive agent. Your options are diuretics, beta blocker, ACE inhibitor, and AT1 blocker. Let me know in the chat box. Let me check the chat box now. AG2 receptors blocker. Okay, let me show you the correct answer. So the correct answer is, it is AT1 blocker. Okay. So AT1 blockers means the angiotensin 1 blocker. I am not sure what do you mean by AG2 blocker. Can you let me know in the chat box? What do you mean by AG2 receptor blocker? I am not sure about this AG2 receptor. What do you mean by that? Can you let me know what is the full form of AG2 receptor? Or were you meant to tell me that it is the angiotensin 2? Yeah, so it is known as AT1 blocker. Okay, your answer is correct. Okay, it is angiotensin blocker, but it is not known as the AG2. It is known as the AT1 blocker more frequently. Okay, maybe it is also known as the AG2 blocker, but AT1 is a more used term. Okay, so yeah, your answer is correct. It is angiotensin receptor blocker. So let me show you. So basically, there are different types of antihypertensive drugs which uses different targets as their method of action. For example, diuretics like thiazides, like hydrochlorothiazide and chlorothiazide, high ceiling like furosemide, clepsydrin, the spironolactone, trimethoprim, and amiloride. These are mechanism of action that acts on kidneys to increase the excretion of sodium and water, decrease in blood volume, and decrease BP. Angiotensin converting enzyme or ACE inhibitors like captopril, linezolid, enalapril, ramipril, and fosinopril, they inhibit the synthesis of angiotensin 2, decreasing the peripheral resistance and blood volume. AT1 blockers or angiotensin blockers are the losartan, telmisartan, valmisartan, and candesartan. They mechanism of action that they blocks binding to angiotensin 2 to its receptor. So your answer is correct. Okay, it is the blocker of the angiotensin 2 receptor, but it is known as the AT1 blocker. And adrenergic receptor antagonists or beta blockers like propranolol, nebivolol, atenolol, mechanism of actions are competitive antagonists that block the receptor sites of the adrenergic beta receptors. Okay, so correct answer was number D, AT1. Now problem number 19 says, which of the following is an example of the water pills? Your options are number A, nebivolol, number B, amiloride, number C, losartan, and number D, captopril. Which of the following is an example of water pills? Nebivolol, amiloride, losartan, and captopril. Which of the following is an example of water pills? Nebivolol, amiloride, losartan, and captopril. Let me check the chat box. I have one answer which says it is number B. Okay, let me check if number B is the correct one or not. So the correct answer is very good. It is number B. Amiloride is known as the water pill. So diuretics are known as the water pills, are medications designed to increase the amount of water and salt expelled from the body as urine. They are often prescribed to help to treat the high blood pressure. Furosemide, sold under the brand name of Lasix, among the others, is a loop diuretic medication used to treat the fluid buildup due to the heart failure, liver scarring, or kidney disease. It may also be used for the treatment of high blood pressure. Furosemide was patented in 1959 and Health Organization essential medications approved for the medical use in 1964. It is on the okay, and amiloride is sold under the name of the Mediamor among the others. It is a medication typically used with the other medication to treat the high blood pressure or swelling due to the heart failure or cirrhosis of liver. Okay. So the last question of today's session, it says which of the following is the correct decreasing order of drug metabolism? Your options are number A, liver, lungs, liver, first the liver, followed by lungs, kidneys, intestine, placenta, skin, and adrenals. Option B, liver, lungs, kidneys, intestine, adrenal, placenta, skin. Number C, liver, kidneys, lungs, intestine, placenta, adrenals, and skin. Number D, liver, lungs, intestine, kidneys, placenta, adrenals, and skin. So which of the following is the correct decreasing order of the drug metabolism? Which of the following is the correct decreasing order of drug metabolism? Okay, let me check the chat box. I hope there will be some answer. Okay, I have one answer which is number C. Let me check if it is the correct one or not. So the correct answer is number A, liver, lungs, kidneys, intestine, placenta, skin, and adrenals. This is the correct order of the decreasing drug metabolism. So pharmacokinetics, as I have already mentioned, is the drug administered, followed by the absorption, distribution, metabolism, elimination. Then from the target of action, that comes from the pharmacodynamics, which is the regulatory proteins, affinity binds to the molecular target, mechanism of drug action, and response to drug, which is the affinity and efficacy. So drug metabolism organ, liver is the heart of the metabolism because its relative richness of enzymes in large amount. Schematic chart of metabolism organ, decreasing order is the liver, lungs, kidney, intestine, placenta, skin, brain, testis, muscle, and spleen. Okay. The correct answer is number A. So this was the last question of today's session. So today we discussed many things. We started with the drug discovery process, various methods of drug discovery and metabolisms. Then we followed we went up to the different antihypertensive agents. We discussed about the blood-brain barrier, ADME, man, also combinatorial chemistry, and so on. So I will share my email ID here in the chat box. feel free to contact me if there is any issue regarding the today's class or any previous class. I will happy to answer your queries. So this is my email ID. If you have any doubt, any query regarding any course materials, so today's class, be it today's class or other class, you can let me know. I will show you once where to find the problem solving sessions. So first you have to go to the SWAYAM portal. In the SWAYAM portal, when you will log in, then you will find there is a announcement chart where you will find the problem solving session in this panel. If you click on it, you will be redirected to a page which looks like this. Here you will find the YouTube links of every class and also a Google Drive folder which has the course content of every class. Okay. So from there you have to find out the week's course content. So now I will close the session for today. Thank you all for joining. We will meet on the next Monday from 6 p.m. to 8 p.m. Thank you all. I will close the session for today.